You are listening to Biotech Hangout, a live and unedited weekly discussion of all the latest news in our industry with a group of biotech insiders. I'm John Maraganore, and my co-host today, really great group of people, are Yaron Werber, Amy Fadia, and special guests Allison, DeAngelis, and Drew Armstrong. Don't you like it that your guys are special, Drew and Allison? I think that's nice.
I wish to be
Um,
special.
yeah. For more information about our hosts and guest speakers or to listen to the most recent episode, please go to biotechhangout.com. Now, Amy, I think I'm supposed to hand it over to you because you also had some disclaimer language to add to all this.
Oh, thanks. Thanks for having me here today. I just wanted to mention that any comments that I make about the companies that I cover are not meant to be taken as any type of investment advice. That's all I had. Thank you.
Yeah, great. And Yaron, do you have anything to add like that you need to?
I do not, but that holds true as well. Thank you.
Great. Okay. Fantastic. Well, we're going to start with some policy topics that emerged during the week. As it's been for the last, 9, 10 months, 11 months, it's been every week has got some new piece of news on the policy or FDA side, and this is no exception to that whatsoever. I think the big news was Rick Pazdur getting selected to and agreeing to become the director of CDER, Center for Drug and Evaluation, which of course regulates the vast majority of products that come out of our industry. So a super critical role by all accounts. Obviously, earlier this month, we saw the departure of George Tidmarsh as the CDER director, and that appeared to be related in part to some of his actions. Related to Kevin Tang and a lawsuit that had been filed. Maybe there were other reasons, but we don't really know. But I think the Tidmarsh departure, sort of opened up the Band-Aid and the wound around industry concerns around stability at the FDA. Which is obviously an area of enormous focus for our industry. We all need a strong and stable and consistent FDA for us to be able to be successful in bringing innovation to patients. So the good news earlier this week, was that Rick Pazdur agreed to step in as the new CDER director. I'm sure everybody on the call has heard about Rick before. He's really a legend at the FDA. He's been there for over 25 years, and most importantly, he is the guy as it relates to oncology. And a lot of the FDA decisions over the last quarter of a century that have come out of the oncology side really owe a lot of thanks back to Rick at the end of the day. So the guy is very well respected. He's a clinician, and obviously a very well known regulatory science professional. So it really is a breath of fresh air for industry, who I think all of us are eager to see more stability and consistency at the FDA. I think we all hope it goes well. There's been some historical evidence that the CBER chief, Vinay Prasad, has publicly disagreed with Pazdur, but there was a very chummy photo that was posted on X just the other day, just yesterday, I believe, with Marty Makary and Prasad and Rick Pazdur. And I think we all hope it's a sign of rapprochement, that these three leaders will work closely together in a collaborative and positive manner to help regulate the science of our industry. Let me see if Yaron or Amy or Drew or Allison, anybody wants to add something to that, but I certainly thought it was good news.
Yeah, maybe I'll chime in, John. So it's definitively good news. Pazdur is one that we all wanted as a stabilizing force. We can all hope for two things. One, that he's going to have sort of the old school but progressive. I mean, from a regulatory perspective, progressive, innovative kind of approach, which will kind of maybe moderate some of the changes and provide sort of an even tone. At the same time, we just want to make sure that ODAC doesn't suffer any setbacks at all, and
Yeah,
that it would be it would be devastating if a year from now Pazdur raises his hands and say it's time for me to move on, which I
Yeah,
doubt.
yeah, yeah. That would be a big loss, not only for Cedar, but now, but also for ODAC for sure. Well, let's go on to some other FDA news, and I think the other big news from the FDA this week was a New England Journal of Medicine article that was authored by Marty Makary and Vinay Prasad on the so-called plausible mechanism pathway. I mean, this has been hinted to by Commissioner Makary on a few occasions over the last 8 months or so. Part of it was inspired by the remarkable story of baby KJ, who is the N of one um, baby who received a CRISPR therapy for a urea cycle disorder. Really remarkable story. And Yaron, I think you wanted to talk a little bit about this pathway first, and then maybe all of us can chime in a little bit about what it means. So why don't you take it away?
Yeah, perfect. So number one, so this came out in New England Journal of Medicine literally this week. It's a very short two-pager, so I commend them on brevity. It's ambitious. It's a little vague, which is okay at this stage, and it sets more kind of an intention than maybe exact policy. Um, Before we dive into it, because there's some interesting things in there, the one thing that's interesting, it is stipulated to go into a bespoke, personalized therapies that are gene therapy and cell therapy sort of focused, but it's got the intention to get broadened to antibodies and small molecules. But the first thing to keep in mind, essentially in parallel, they decided to immediately sort of a little bit go against it in the Unicure situation relating to Huntington, where at the end you can argue that some of these elements would apply to Unicure, and that was sort of set in motion. We talked about this last week in detail, and now they seem to are going against what was already agreed on with the previous team. So this is basically you need to have a biologically defined cause that is pathogenic with a specific mutation or target, characterized by natural history. You need to show that you can successfully engage and target it. Specifically, they're talking about gene editing or mRNA sort of therapies. You could use animal or non-animal models if applicable, and you could do invasive or preferably non-invasive methods for confirmation. You need to have a clinical outcome that's consistent with improvement when there's progressive deterioration, or a disease-free course if the disease waxes and wanes. And the FDA will even consider the patient as a natural history, as their own control. The goal is to even have single patient sort of expanded access to generate sufficient data to support marketing approvals. This is where now it starts getting a little vague and sort of aspirational, that they're saying that even if you can start with one patient and have several consecutive patients that are successful, that could lead to marketing authorization. Then they talk about you can use that as a platform technology for a personalized product to support similar therapies and other conditions. Exactly what that means, it's unclear, and you can support potentially accelerated or regular approval. And then, of course, there's post-marketing commitments to look at safety, as you know, and risk mitigation. But where it's a little confusing is exactly how broad can one patient really lead to approval? What do they mean by few patients? What do they mean by platform um, to then lead to kind of broader approvals? And they even mention a situation where, let's say, you have a disease with 150 mutations, is the example they're giving, that ultimately have a common symptomatological manifestations that's addressed with this therapy, that it could lead to approval for all mutants. So, well, let's see. This is certainly is a positive development, but let's see how they implement it.
Yeah, I think that's a great summary, Yaron. I also wonder if it's for the baby KJ examples, very narrowly defined, and as you know, there are many N of one efforts that are out there. I'm involved with NLORM, which is the effort using antisense oligos that Stan Crook has pioneered. There's also another couple of groups out there that are really looking at N of one type of tailored therapies. I think the scope of this plausible mechanism, aspiration is something which I think remains to be seen. It'll be probably really important to see how it gets written into guidelines ultimately to understand, what the breadth and the scope is of all this, and obviously, does it apply to a unicure like situation? That certainly seems at odds to what was written here, in terms of what we all understand. So I think we'll have to see how that gets defined into guidance. I don't know if others, Amy, Drew, Allison,
Yeah,
do you have anything to add?
I think I could sort of, I kind of agree with you that, this is really positive in the sense that it encourages innovation, but how it gets implemented is really going to be important because if it's, used inappropriately, then it could lead to, safety or efficacy failures, and maybe that could sort of put a setback to what, this is intended to be. And I'd also be curious to understand how payers would view this and, how much of evidence that they would need to see, and how pricing gets determined based off of that.
Well, I think that's a super important point. Obviously, even with accelerated approval pathway drugs, there's always been the question around any implications on the payer side of it. And so, that'll have to be watched pretty carefully as this thing gets rolled out. There's no doubt about that. Anything from Drew or Allison on it, or
I wanted to, yeah, I wanted to echo something that and expand on something Yaron said, which is just this idea and this theme that I feel like we've seen over and over again this year, which is that there's a headline policy that people really seem to like and are excited by, and then the actual execution from a regulatory standpoint seems to like run counter to that over and over again. And so, you know, people keep looking for, that I chat through to are looking for some kind of consistent regulatory philosophy or approach, and it keeps kind of being contradicted by their own actions. I think that's starting to really get to people now that we've seen a couple of repeats of it, and it's starting to create some, I think, meaningful concern that we might not get to like a really stable, consistent path forward on some of this stuff, and some doubts when we see these new policies roll out.
yeah, I mean it's a super valid point, Drew, and there's been some Wall Street Journal editorial board op-eds on this exact topic, and I think we're all looking for some consistency, on regulatory guidance and action. It's super super important at the end of the day. Maybe we can segue to one of the other big events that happened this week, and that's the MAHA Summit. So, Allison, I think you covered it very nicely, but I'd love to hear what you've learned about that gathering and maybe provide some context for the call.
Sure. Yeah. So we started hearing Tuesday morning that hundreds of people, including members of the regulatory body, NIH, FDA, JD Vance, RFK Jr., Secretary Kennedy, and members of our biotech community were going to be meeting in DC to have the inaugural MAHA Summit. There was a schedule that we were able, myself and my colleague Daniel Payne, were able to get our hands on after Politico was kind of the first. I should give a little hat tip to them to disclose that this summit was happening. And taking a look at the documents that we were able to get, the invitations, the agenda, it was really pitched as, exclusive gathering of kind of people in the know in various sectors, mixing and mingling and networking. And we saw a whole series, there was a whole day of events, of sessions featuring, some of our the people that we know well in the biotech industry over at, CRISPR. Sam Kulkarni took the stage along with Alexis Borisy and George Yancopoulos, where I understand that baby KJ was a topic of conversation, and also, Sam talking about what CRISPR is able to do to address and really treat very efficiently sickle cell disease. Uh, that took, kind of was center stage. Saw some conversation about oral GLP-1s hitting the market, Dr. Oz said that that could happen as early as March. And it's worth noting that Eli Lilly's oral GLP-1 candidate, orforglipron, recently received one of the commissioner's vouchers for review, for expedited review. And interestingly, I think this was a notable absence from what we were able to gather from people who were in attendance, was that vaccines really weren't much of a topic. It was really, I think, focused a little bit more on kind of general food, lifestyle, general health trends, even some brain computer interface conversation. Apparently, there was a panel with Neuralink, with the CEO of Neuralink involved that got some cheers. And it was really a full day summit on Wednesday where, kind of MAHA, I would venture kind of took a little bit of gave themselves a little bit of a pat on the back for some of the work that they've been doing and talked about their vision for what's to come and handed out some goodie bags with some, beef tallow potato crisps.
Allison, were you actually there, or was there any media coverage, presence at the meeting, or was it
So
closed
this was
media?
interesting. It was a closed media event, invite only from what I understand. And I say that not even just that, members of media organizations that the administration might not like weren't invited. No, essentially no media was invited, even,
Mm.
Fox News and some organizations that, the administration likes. The one exception was Vice President Vance's he gave a fireside chat kind of midday, right around lunchtime. CNN had like the pool coverage that day, which means that it was kind of their turn to have first access to cover what Vance was doing and talking about. So his JD Vance's session was broadcast. That was the only session. So everything that we were hearing about was from people on the ground. And I do have to say that one of my colleagues, Chelsea, was in the venue. It was at the Waldorf Astoria down in DC, and she was in kind of like the lobby, sitting at the bar, trying to scope out what was happening live on the ground. And she said it was a very interesting meeting to observe.
Oh, fascinating. Well, look forward to my MAHA Summit 2 invitation in the coming years.
So did you
Not.
not get invited
No,
to
I
Summit
didn't get,
One?
Allison. I wouldn't have gotten invited. I didn't get invited, so it's all good.
Interesting.
Well,
Well, good. Let's.
I can tell you it's been very successful in the sense that now I'm actually looking up what are these chips, and I might
Oh,
actually even try them. So at least they're going to get maybe one sale out of this.
that's
I've been
great.
told that that they taste like normal potato chips, to be quite honest.
I love it. Well, fantastic. That's really helpful, Allison. Thanks for that coverage of that event. Let's move on to some data and company news. And I want to start with Alkermes, and there's also some interesting business deal news on Alkermes as well. But let's start with their Vibrance 2 data for their orexin agonist. Amy, do you want to cover that
Yeah,
for
sure.
us?
Yeah, so this week we received the much anticipated Vibrance 2 data from Alkermes for their orexin 2 agonist in NT2, which is narcolepsy type 2. And this was the first robust data set in this population with data in over 90 patients. Earlier this month, Centessa had reported data from their orexin program in NT1, 2, and IH, but it was from a relatively smaller data set. Now, the Alkermes results now provide kind of a strong evidence that the orexin agonists can deliver meaningful clinical benefit beyond just NT1 in a heterogeneous population like NT2. And, this also strengthens the broader thesis around this class and the applicability in other larger neurologic populations, such as, beyond just IH, but also into MS and Parkinson's. Certainly, at least builds towards that. Now, there was some debate about kind of how this data stacks up, and there was some cross-trial comparisons that were being done, which to some people may have suggested, a lower placebo-adjusted improvement in MWT for the Alkermes data relative to what we had seen from Centessa. Now, Vibrance 2, in Vibrance 2, Alexorexton achieved a placebo-adjusted change in MWT of 9.3, 6.7, and 6.7 minutes for the three doses they tested of 10, 14, and 18 milligrams at week 6. And in comparison, the Centessa data had shown over 10 minutes of change at week 2. Now, while I think that it's expected that people are going to compare data across trials, but I think there are two caveats that I think are worth mentioning. Firstly, it's sample size. Alkermes had enrolled 93 patients, whereas from Centessa, we got the data from 10 patients. And given the heterogeneity in the NT2 population, I think that a larger data set probably is critical in really fully characterizing treatment effect. And then secondly, there's treatment duration. The Alkermes data had been reported at week 8 versus week 2 for Centessa. And at this point, we don't fully know how long or how much of tachyphylaxis emerges and how long it takes to reach steady state. And so I think we're going to need to continue to sort of watch how these data emerge over time. The other key efficacy endpoint was ESS, which better captures the patient's subjective experience of sleepiness and probably is a more relevant regulatory endpoint, especially in EU. And at the two higher doses, Alkermes showed, roughly around 10-point change, or patients going down to 10 points in ESS, which is where, sort of patients are considered as being normal. And this was sort of comparable to what, Centessa had shown at week 2. So I think, it'll be important to see how these data evolve over time as these companies continue to sort of dose escalate and pursue higher doses and split dosing. And interestingly, on safety, we saw a very consistent on target profile. The main adverse events were insomnia and urinary urgency. And importantly, visual disturbances were less frequent than what we saw with the Alkermes data in NT1 population. And what this means is that perhaps in NT2, it allows these companies to continue to explore higher doses and potentially also explore split dosing strategies. And we're seeing both companies, talk about doing that. So overall, I'd say that, this class continues to remain highly compelling. We continue to expect, I think, multiple companies to reach market, although it's still a little early to know if there's any one player that'll dominate the market. And perhaps, the real differentiation could come from other endpoints like secondary endpoints around cognition or nighttime sleep. Maybe, we need to look at how the dosing flexibility ends up, and maybe ultimately it's time to market and payer dynamics as well.
Yeah, that's great. Helpful, Amy. Do you want to comment, since we're on the topic of Alkermes, do you want to comment on the competitive situation with Lundbeck making a $2.6 billion bid for Avadel, which had previously been announced to be acquired by Alkermes for 2.1 billion? What are your thoughts there?
Yeah, I think it's interesting, and I think we are seeing this is sort of a second recent case that we are seeing where there's bidding. And I think overall it's good to see M&A heating up. I think in the case of Avadel, it's interesting to see how the deals were structured. I think in the Alkermes deal offer, I think they had an upfront payment of 18.5 with a CVR of $1.50 linked to approval of idiopathic hypersomnia indication for which they're going to have phase 3 data next year. Whereas the Lundbeck deal structure, there, out of the $23 includes $2 of CVR, which are linked to sales performance milestones at 2027 and 2030. So it'll be interesting to see how this evolves. Alkermes sort of immediately put out a press release this morning that they're considering their options. I'd be surprised if they don't come back with at least a counter offer. And perhaps, there's opportunity even within, the total deal value of $23 to improve the attractiveness of the deal. So yeah, I think this is an emerging situation, and
Amazing.
we're going to see more come through in the next couple of days, I think.
Yeah, and amazing that this is now the second recent example of a interloper coming in after announced deal, and we'll talk about Metsera in just a minute. But do you view, Amy, do you view any potential more limited FTC challenges with Lundbeck versus Alkermes, given the orexin program that Alkermes has? Is that one angle that Lundbeck has articulated?
You know, it's certainly a possibility, but I mean, these are completely different mechanisms,
Yeah,
and um, I don't know if that necessarily, one is a scheduled drug, the other will not be. One is nighttime, one is daytime, and then there's sort of a broader opportunity with the orexin platform compared to the oxybate, and particularly with there being competition in the oxybate market, and there are going to be additional generic versions of Xyrem that are going to launch early part of next year. I don't know how much pricing power in the oxybate market Alkermes could have simply because they've got an orexin as well.
yeah,
So I don't know if there are, aside from, yeah, they're both targeting sleep. I don't know if
yeah.
it truly gives them more pricing power.
Yeah. Okay. Well, let's then move on to the other recent competitive deal, and Drew just this morning wrote a really fantastic story about the Pfizer Metsera acquisition, and as I'm one of the original people that was involved in helping Metsera get started, so I I thought Drew's reporting was great. Drew, do you want to comment on that at all?
Yeah, this was if you haven't read the story, it's on our front page over at Endpoints. It's worth checking out. I think, we cover deals all the time. I'm always interested whenever we get a look inside the boardroom and the high intensity negotiations that are going on, because I think, maybe a lot of people on this call have been able to be part of those, but for a lot of folks who work in biotech, this is really a closed door environment. I think it told us, though, a couple of key things. I mean, I think one, we learned a lot about Novo's motivations, and this was a case where we had two big companies that really needed to find out how to get their vibe back. Essentially, Pfizer's been looking for its post-pandemic move for a long time. And they'd had a failure in obesity. Novo had really lost the obesity early lead and the crown there. And so you had two companies that really, really, really needed this. And I think that's what's fascinating, to just see how aggressively they were going after this particular asset. I think it leaves us with a great question about what Novo does next, because nothing about their situation has changed. I think some folks may have seen Pfizer taking a small bit of additional revenge on them this morning. Pfizer's former head of R&D, Michael Dolston, was supposed to join the Novo board and get voted up to that today. Last second, he pulled his own nomination, and our understanding is that Pfizer raised some objections about that. So this was kind of a deeply nasty and, at times, personal almost fight between these companies. I think the other piece of this that's really interesting, and Amy alluded to some of this, we've all been waiting for a really long time to see if we were going to get a good first look at how the Trump administration is going to approach antitrust in biopharma. I think we caught a glimpse of that here, but in some ways it's tainted by, I'm going to speculate here, but maybe tainted a little bit by Pfizer and Borla's close relationship with this administration, which they were really talking about. I don't know how heavily they were actually able to lean on this, but if you look in the proxy documents, the FTC called Metsera on the final day of this. This was November 7th. Basically said, if you try to do this deal, we're going to sue. And so I think we got close to getting a real glimpse on this, but they were more focused, I think, on the structure of the offer and the kind of unusual way of putting this transaction in place rather than actually the anti-competitive aspect. So I was hopeful there. I think we still don't know, but I do think it is something that's going to be interesting to watch closely if we start to see more M&A happen around the space. And I will also just highlight our cousins over at the Financial Times. They had some reporting last night, I believe, about the Merck Cidara deal, suggesting that there was another bidder up until the very last minute in that transaction as well. I don't know if we see something go public, but it seems like all of a sudden, we're moving away from single bidders, CVRs to get things across the line into competitive and at times nastily competitive situations. So I think, the last 2 months of the year, last month and a half of the year are going to be fascinating from an M&A standpoint.
Yeah. Well, I mean, it just highlights the intense interest from pharma in biotech assets and probably recognition that, this is the time to either buy them or lose them at the end of the day. Why don't we then, just because you mentioned it, Drew, why don't Yaron, do you want to talk about Merck Cidara? Because that just happened this morning. That was another big piece of news, yet another $10 billion acquisition, which is
Yeah.
fantastic.
Yep. So Merck is continuing their recent playbook. What they're doing is continuing to build up their cardio, in this case, really pulmonary capabilities, with a late stage asset. It's got breakthrough status with a strong phase 2b um, to help the Keytruda LOE, which we all know is 2028. So Cidara is getting bought for 9.2 billion. It's 3x their recent market cap. And again, remember, Merck is increasingly going into pulmonary with a recent Verona deal, obviously the previous Acceleron deal. Closing is expected in the first quarter of next year. So what are they buying? They're buying a drug called CD388. It's a long-acting strain-agnostic antiviral. It's a neuraminidase inhibitor, but it's tagged to a proprietary Fc fragment. So it's essentially creating a long-acting, low molecular weight biologic with an antiviral for a once disease season preventative for influenza A and B, so seasonal and pandemic. The phase 2b looked really good. It showed a 60 to 76% protection against lab confirmed flu with a single shot. So recall typical seasonal vaccines are around 40 to 50%. Phase 3 is ongoing, and the data is expected. This is the anchor study in high risk adults and adolescents. The data is expected in the first quarter of next year. So, yeah, the data the phase 2b looked really, really good. And so it's nice that Merck continues to be fairly aggressive and active.
Yeah, absolutely.
I'll just
Well,
add
look, I mean,
on.
go
Oh,
ahead.
sorry, John.
No, go ahead, Allison.
I was just gonna add on to expand a little bit on that. We had Dean Lee, one of the executives at Merck, their head of research, on our podcast at Stat the Read Out Loud this week, and we were asking him about kind of this precise issue of how is Merck looking to the future, uh, kind of post Keytruda and that patent cliff that they are facing. We were talking in particular about their work in heart disease with their PCSK9 oral medicine. But Dean was indicating that Merck's strategy is not going to be, kind of searching for one big product, that they are looking to do an assortment of deals and have an assortment of products that they will bring into the company over the next couple of years to help kind of backfill some of that patent cliff and then even grow on that.
Yeah, it's fascinating. Well, it's good to see. It's good to see these transactions. We had another very interesting deal this week, and I can sort of tee it up and then see if other people have comments on it. But it was the acquisition of Mersana by Day One, Dawn. And what's unusual is it's a relatively small size, mid-size, barely mid-size biotech, Dawn, with about $1 billion market cap, buying Mersana, which had been, challenged, I think, in its story, but certainly one of the pioneers on the ADC side. But they went ahead and they put an offer to acquire and was agreed upon Mersana, and largely for its B7H4 ADC. I mean, for Dawn, for day one, it obviously is an effort to build on, their first commercial product, which is Ojemda, which is an oral brain penetrant RAF kinase inhibitor, which showed stunning data in pediatric low grade glioma, really terrible indication, but relatively small market, of course. And in addition to that product, they do have their own ADC efforts. So they grabbed Mersana and, obviously are consolidating that effort within Dawn. But it's not usual for us to see smaller biotech acquisitions taking place. So that was, I think, a bit notable in this one. I don't know if anybody had any other observations or thoughts on that deal to follow up on.
I think I could add that I think it was interesting also how the deal was structured. I think it's sort of giving kind of the mid-sized or sort of the smaller biotechs the ability to acquire and build their pipeline with a low upfront, and majority of the deal value was is linked to development and regulatory milestones. So I think it allows day one to get access to this drug without paying a huge amount upfront. And the other thing that was interesting is I think it's a differentiated asset. There are a lot of companies that are pursuing B7H4, and but a lot of them are really going after ovarian cancer and endometrial cancer, and that's probably where Mersana was also expanding the cohorts. But the main anchor of the deal for day one is really the ACC indication, which is sort of a rare, cancer in the salivary gland, where the expression of B7H4 is actually quite high, around 94%. And the data there was actually quite compelling, where they showed about, 55.6%, response rate in that subset. So I think it gives day one um, a unique way to go after a rare disease, even though, as such, the B7H4 market is pretty crowded with a lot of large pharma pursuing other indications with it. So I thought that was
Yeah,
interesting.
yeah, that's super interesting. Well, why don't we move back to some data readouts from the field, and Amy, if you could maybe comment on Neurocrine's unfortunate news about their MDD program, that would be a helpful place to start.
Sure. So yeah, on Monday, Neurocrine announced its phase 2 data from its signal-seeking study of NBI 770, which is a negative allosteric modulator of NR2B subunit of the NMDA receptor. It was a 73 patient study in MDD and did not reach its primary endpoint. The company did note in its press release that there are some aspects of the data set that warrant further analysis before determining next steps. Um, the interesting thing is that the broader NMDA pathway has been long pursued in depression, especially with the strong and rapid activity we've seen with ketamine and esketamine. But the mechanisms are different. With ketamine, it involves a broad blockade of the NMDA receptor. Whereas, with this particular drug, it's sort of targeting kind of a subset of that. And so and then sort of the goal is to sort of spare PV plus interneurons, which cause the dissociation effect that we see with ketamine. But it seems that maybe this is sort of failing to trigger the full circuit level changes that are required for the robust antidepressant activity that we would have liked to see. And we've seen a long history of failures in this space. And so with this data set, the question is, well, maybe that's the reason that we are just not seeing the broad activity that is needed around AMPA, mTOR, BDNF that you need for antidepressant, or um, whether maybe this was just sort of a small multi-arm study that was sort of underpowered to see the rapid effect at day 5 that they had designed it to. So I think we're going to need to see what, the company sort of reveals after further analysis. But, for the time being, I think the outlook on this mechanism is, probably less attractive.
Such a tough space and enormous unmet need. I hope we can catch a break one of these days, no doubt about it. Let's turn to some news from the American Heart Meeting, which happened this past week and over the weekend as well. It was actually pretty um, action or data filled meeting by AHA comparisons. And one of the readout that was really quite interesting was the CRISPR program with ANGPTL3. Yaron, do you want to comment on that a little bit?
Yeah, absolutely. And maybe just to throw in there, and I'll touch maybe quickly. Ionis had really good olezarsen
Yeah,
SHTG data, and of
they
course
did.
Amgen had the Vesalius, right?
Yeah.
CV primary prevention. So let's start to your point on CRISPR. So CRISPR showed data with their Eng PTL3. It's a Cas9 in vivo. 15 patients with high cholesterol, high lipids, and mixed dyslipidemia on maximal background therapy, single IV infusion. Data looked really, really good. And it's really encouraging. And again, this is also relevant to what Ionis is doing. So ANGPTL3 is trying to reduce dyslipidemia by having the muscle and other tissues outside the liver actually do the metabolism, which is different actually than what Ionis and Arrowhead are doing. So it showed a 73% reduction in PTL3, dose dependent. So it was nice to see that. A 55% reduction in lipids, almost a 50% reduction in LDL, 33% reduction in ApoB, and about a 50% reduction in non-HDL. And participants who came in with sort of lower, above 150 levels of lipids showed a 60% reduction. So really, really good overall. And then if you look at people who actually had a PCSK9 inhibitor, they got sort of upwards of more than 80% reduction in LDL. The big thing, and I think John, you're gonna want to. It sounds like you're gonna tie. Later on, we're gonna talk about Intellia and the patient issue that died
Well,
there.
why don't
It
we just merge those? Why don't we just merge those together,
We can
Yaron?
merge them. Yeah. So safety looked okay. I mean, there was no treatment-related SAEs, no grade 3 liver enzyme elevations. 20% had grade 2 reversible infusion reactions. One participant had grade 2 transaminitis resolving by day 14, but there was one death, which was reported not judged to be related to treatment. They are planning now on moving into a phase 1B. Was published in the New England Journal, and New England Journal is calling for a 15-year, obviously, in vivo genome editing sort of surveillance. So again, data looks really good. You got to put this in context because you can take an antibody from Regeneron, and you can take other ASOs or siRNAs, um, which will require obviously more continuous dosing, frequent dosing, not one and done. But let's discuss now Intellia. So Intellia, this just literally this week and 2 weeks ago, had the unfortunate situation. They were in phase 3 for ATTR cardiomyopathy, something very close to John's heart. This is a CRISPR based therapy. So they actually dosed over 650 patients in the magnitude 1 and 2 studies before less than 1% had grade 4 liver enzyme elevations. In magnitude 2, that only enrolled about 47 patients, and they didn't have any. So what happened? Well, in late September, sadly, one elderly male in his 80s in the first magnitude died. He basically was hospitalized with grade 4 transaminitis, increased also bilirubin increase, clearly markers of liver injury. And then a few days later, this was announced on October 27th. At that point, they went in clinical hold on the 29th, and they announced that the patient sadly passed away it looks like around November 5th. So this program is now on clinical hold. And recall, Amvuttra, which John developed, is an incredible product, Q3 months dosing, approved now, based on the Helios study. And And of course, Ionis and AstraZeneca are behind, and they're going to read out their CardioTransform study probably in Q3 next year, also for cardiomyopathy. That's a outpatient sort of auto-injector monthly. So it really begs the question, do you really need a CRISPR-Cas9? And sort of what's the venue by which this comes back at this point, you know? It's going to be
Yeah.
a big overhang.
Yeah, it is. It is a challenge, and obviously the CRISPR ANGPTL3 data and the death, while graded as unrelated, it's odd that that would be happening in a population like the initial population they aim to study. So I think we're going to have to learn more to understand more at the end of the day. I do want to talk on a sort of related, but not totally related topic, which is the work that Coro reported this week on their alpha 1 antitrypsin deficiency program. Their effort is focused on RNA editing, which is a reversible approach. It harnesses the 8R enzyme in cells to achieve base editing, we in 2024, there was some very promising data from Wave Life Sciences on alpha 1 antitrypsin base editing, which they updated a little bit recently, which looked a little bit more confusing. But Coro chose an LNP based approach, and it had very promising preclinical data, but it did not meet the thresholds. In that study, in their rewrite study, which is a phase 1/2 study that they did, and they've chosen, I think, smartly to discontinue the program in light of not only the data that they generated, but also the competitive landscape. Because of Wave and also other players out there, the company called Irna, as well as what Beam is doing with base editing at the DNA level. But I think they made the right decision, the tough decision. They got penalized as a stock, but we're going to see if they can pull it all together, which I hope they do with their program in urea cycle disorder. But that was also some other news. And then maybe Amy, you can quickly cover because we're running out of time a little bit, the Cogent news in gist with those programs.
Sure. This fall, it seems like we're seeing several companies double their market cap at the back of positive data readouts. Earlier last month, we saw Praxis in my coverage universe do that, and this week it was Cogent, where they delivered standout data set in GIST. The first line therapy in GIST is Gleevec, which has, a PFS of almost 19 months, but the response quickly goes down significantly once you hit second line. And it's sort of the standard of care in second line is sunitinib, and has the PFS of about a little over 8 months. So what Cogent did was combined bezuclastinib with sunitinib, and what bezu does is it offers a complementary mutational coverage. It targets KIT exon 17 and 18, whereas sunitinib targets exon 13 and 14. And in the phase 3 peak study that they announced, they reported median PFS of 16.5 months, which is pretty impressive relative to about 9 months that was shown by sunitinib, and an overall response rate of 45.6% versus 25% for sunitinib. Now, I mean, the enthusiasm is warranted, particularly given, the history of GIST. It's sort of been a graveyard for targeted oncology programs, where several drugs that had promising data in phase 1, 2 ended up failing in larger trials. And one I can, obviously seems recent is Deciphera's Quinlock drug that failed in their phase 3 INTRIGUE study, and then there are a couple others in the past. And from a commercial standpoint, this is sort of a 4 billion plus market opportunity, given about 3,000 patients that are in second line. And following the data, it was good to see Cogent's market cap increase, over about 2.5 times, and they were also able to raise capital at the back of that with a $200 million equity offering and $200 million convert. So I think it's just good to see positive data and companies being able to, capitalize themselves to continue to sort of develop programs in the future.
Well, it's certainly a sign of the markets coming back, which we're also saying more generally with the XBI when companies can get rewarded for good data and robustly raise capital as a result of it. There, a couple other topics we want to just cover, and then we'll do a wrap up. I mean, we did see this past week. Actually, it was I believe Friday last week, but perhaps after the last hangout was recorded. The passing of James Watson. Obviously lived a very long life, age of 97. He was the co-discoverer of DNA, the structure of DNA, along with Francis Crick, and he's really the last of the key scientists who were involved in the discovery of the structure of DNA to have survived. I mean, that includes Watson and Crick, of course, but Franklin and Wilkins and even Linus Pauling. So it is the end of a generation of scientists that serve as the foundation for most of everything we do. I mean, his remarkable scientific career, unfortunately, was just clouded by a history of unacceptable comments that were both bigoted and prejudiced, and he was ultimately shunned by the scientific community because of his behavior, if you will, which I think appropriately people found unacceptable. So sad to see him go, as with any human being, but unfortunately, his history was a little bit challenged, I would say. I actually had the chance to meet him once, and he knew about Alnylam. He knew about our efforts of advancing RNAi, and the one thing he said to me was, "John, just go fast." It was good advice, I guess. So anyway, R.I.P. Jim, and I hope you find peace in your next chapter. The last thing I want to cover is something which was fascinating this week, which is a podcast with Dave Ricks on Cheeky Pint, which I had never heard of before. Was This was brought to my attention. It's a podcast that's done by John Collison, and I haven't listened to the full 2 hours of this podcast, but I listened to a good chunk of it, and it's actually a really remarkable interview that covers a far-reaching set of subjects from R&D to glitz to drug pricing. Um, and I was really struck with how amazingly well and authentically Dave Ricks explains our industry in this podcast. So I hope there's more of this because it's so important. Podcast is such an important way to speak to the public these days. But I just also was really proud of Dave and how well he did this. I don't know if any of the other speakers on the hangout here listened to this as well, but I'd love your thoughts if you did.
We were definitely chatting about it on the Stat Slack this week. Particularly, what was the line that he had about, if Taylor Swift can
Right.
sell out X number of arenas, we can certainly enroll X trials.
Yeah.
And
Yeah.
as someone who has like we've had Dave Ricks on the podcast at Stat, he is a very good public speaker.
Yeah.
And he clearly has tapped into the fact that he needs to be speaking to the consumers, not just to kind of his peers and people in healthcare, but to the consumers. And I found it really interesting also because I had had a conversation recently with Kathy Friedman over at GV, and she was talking about how, her view of what's happening. She's like, we are in an era where, healthcare is becoming more and more consumerized. And so I think that, the type of stance that Dave took to go on a podcast that is, it's not for you and me and Drew and Yaron and Amy. It's for the average person who may not know how our ecosystem works. And, to speak so candidly was really a smart move.
Well, I'll tell you, I would, like I said, I would love to see more of it. because we need to connect with the broader public in better ways. So we all know that we as an industry, we've done a horrible job communicating what we do and why it matters and why it's good. And I thought that was a really terrific start from Dave. Um, well, look, we've only got a few minutes left. I want to leave some time for people to make some final comments. So let me just go around the horn here. Looks like Yaron has stepped off. So let me go to Drew next. Drew, do you want to
Yeah,
anything to add?
when you guys were talking about all these, something I've been thinking about a lot, looking at the data coming out of AHA, it's just fascinating to see this explosion of new potential treatment modalities in, cholesterol. I mean, we've gone from like statins to having a, long-acting injectable PCSK9 biologic to now potentially these gene therapies, and then now from Merck potentially a small molecule PCSK9. I mean, if you're treating high cholesterol, or are a patient, I think it's pretty extraordinary. I think it's going to be fascinating to watch the kind of commercial and scientific battle, to see where these things find their place in the world, and absolutely watching, I think, what that suggests about other diseases where we may see a lot of different approaches where you know efficacy doesn't always have to come with more complexity. Sometimes it will, but it's really, really cool to see, and I can't wait to see that battle between methodology mechanisms start to unfold.
Yeah, yeah, I think that's right. Amy, anything to add in
I
closing?
yeah, I heard a little bit, maybe about half of the interview of David Ricks, and uh, I think I would recommend everybody on this podcast to also listen. Some of his comments about how to drive transparency in pricing while also not adversely impacting the amount of effort that goes into research to develop new medicines, and how do you sort of solve for that? And he sort of, laid out a couple of ideas that he had. I thought that that was really interesting, and that, gets to the core of what, is happening at the Hill, and, what I think a lot of us sort of think about day in, day out.
Yeah, I agree, Amy. Allison, any last comments before we wrap up here?
I can't think of too much other than that, we're looking to see who the next players in a bidding war are going to be,
Yes,
because we're
that would
certainly
be
seeing
fun.
more M&A.
Yeah, yeah, you bet. No, well, I mean, let's see if this Merck deal, has a bidder that comes in, given what the FT was reporting. That would be super exciting for sure. Well, listen, everybody, thanks for taking the time. Have an amazing weekend, and we look forward to being back next Friday and chatting with all of you about our amazing industry.